Hello everyone and welcome to Biology Made Easy. In this video I'm going to discuss about the genetic material and its forms inside the cell. So we are speaking about the eukaryotic cells. Eukaryotic cells have nucleus, and inside the nucleus we have the DNA. DNA condenses inside the nucleus to form structures called chromatin. Chromatin is a very random structure of DNA, but when cells want to divide, the chromatin will condense and wrap around proteins in order to form the chromosomes. So chromosomes are organized and condensed structure of DNA that are needed for cell division. In addition, we have within the nucleus the nucleolus that includes DNA, ribosomal DNA that is needed for the production of the ribosome. So this is how it looks like inside the nucleus. We have the chromatin, we have the nucleolus, and all of this is surrounded by a membrane called the nuclear membrane. To understand more about DNA, DNA is a double helix, and inside the cell we have huge molecule of DNA, 3 meters long. So in order for this long molecule of DNA to be inserted inside the nucleus of a cell, it has to undergo certain rounds of condensation or packaging or packing. So the first round of packing is it will coil around proteins called nucleosomes. It will fold or coil around proteins to form nucleosomes, and these proteins are called histones. So histones are proteins that help in the process of condensation of the genetic material. As a result, the structure that is formed, it's a random. We call it the chromatin. So this is how DNA looks like in cells that are not divided. So cells that are not dividing, their DNA is arranged as chromatin with a dense region inside the nucleus called nucleolus. And this nucleolus, it contains DNA that are non-coding. They don't code for any production of the protein. On the other hand, they code for the production of a certain type of RNA called ribosomal RNA, and it is responsible for the production of ribosomes that are the factories of proteins in the cell. Okay, so if you want to write down the 3 forms of the DNA, we can write chromatin, and chromosomes. That are made of 2 chromatins. It's very important to differentiate between these 3 terms: chromosomes, chromatin, chromosomes, and chromatin. So what is chromatin again? Chromatin is the DNA which is not organized, and it is located inside the nucleus of cells that are not dividing, and they are usually visible when the cell is during the interphase stage, which is the preparatory stage of division. Now, when the cells want to divide, having chromatin won't help for the cell to divide. As a result, this random distribution of DNA must be organized, and it will be organized to form what we call the chromosomes. So the chromosomes are well organized structures that are made of two strands or two parts. And the reason why there are two strands is later on they are going to separate. So if we take this chromosome, when we look at it, it is made of two parts. These parts are joined in the center by centromere. It's where the two strands are attached, and every strand in the chromosome is called a chromatid. So this one here, this is called chromatid, and this one here is called a chromatid. And in our cells, when our cells want to divide, the chromatid will form 46 chromosomes or 23 pairs of chromosomes. And these two parts, I'm going to label them in red. These are called here at the edge of each chromatid, it's called telomere. So there are two telomeres that protect the end of each chromosome. This is a chromosome, and chromosomes are visible in cells that are dividing. So when this happens, this is called the condensation of DNA. This happens when the cells want to divide. Because when the cell divides, it cannot split the DNA if it is randomized like that. So the only way cells can split the DNA after copying it in interphase is to form the chromosome structure. Okay? So if I'm going to draw a cell which is preparing for division, this cell at the start of mitosis is going to have chromosomes and these chromosomes can be visible during the second phase of mitosis, which we call it the metaphase. So, if we look at a cell with visible chromosomes, we can tell directly that this cell is about to divide or it is dividing. Whereas a cell which is not dividing will have this. And this is called the chromatin material. Okay, so this is called the chromatin, which is made of DNA and proteins, but they are not condensed. And those are called the chromosomes. It's the same genetic information, but here the chromosomes are arranged. And as we said, each chromosome is made of two chromatids. So this cell didn't start dividing, this cell is divided. Thank you so much.